And that's more efficient. And this is what people want from the American universities right now, is them to go faster. They don't want them to take years, because it's wasting people's time, giving them courses that they really don't need. And so, Dr. Usama, how are you going? I see your presentation now. You are ready to go.
Yeah. Should I start now?
Yes. Please go ahead.
Okay. So we are here to discuss about the biostatics and biochemistry, mainly the scope of these two subjects, and I'm going to correlate it with each other and discuss its prospects and the its importance in the further career if you choose it the biology. So going with the introduction, biostats and biochemistry are closely related fields to the study. If we go down the main branches of the biology, biochemistry is basically the scientific study of chemical and molecular processes, and biostat is sort of a technical thing in which mainly we deal with the data. If we are talking about biostat, we are talking about the biological data we are dealing with. We mainly do sort of experiments, record our data, manage our data, and analyze and interpret our data in biostats. So biochemistry and biostats provide insights into the complex interactions between molecules, cells, and organisms. Let's start with the cell. You may already familiar with all of you, the students. If we start from the cell, suppose we want to develop a drug, we basically start from the normal structure of the cell, right? We will learn about how cell works, how cell is made up of, its structure, its membrane receptors, and each and everything. First we will learn that in the biochemistry, right? Then we study about its pathology. If we're talking about a disease and trying to develop a drug against that specific disease, we will go for a structure that fits with the structure of the cell. We also deal that part in biochemistry. Suppose we develop a drug, a sample drug, then we want to try it on some people, sort of a control or volunteers. Suppose if some volunteer is for the if that drug is for cancer, for example, we want to try it on ongoing research, sort of in cancer patients. Then this part, right after the trial starts, it comes into the biostats. We choose a control group. We choose different type of people with different ages and genders. We apply, we introduce the drug into them, then we check their results, we record their data, we manage their data, we analyze with different tests in biostats. Then we have a result at the end. We share that result as sort of in research, or we implement that this drug is good to go, or is good to it's good for the patients, and we can finally let it out in the market. So these two areas of biochemistry and biostats are more like interrelated if we talk about them, and their scope is way more diverse. Speaking of biostats, we are going to dig deep into it. So if anyone has any question, please ask me at the end of the topic. Okay, biostats. It is defined as the application of statistical methods to biological and medical data. It is a component of many areas of scientific research, including genetics, epidemiology, and medical diagnostics. So basically, biostat is a very close subject of public health. We mainly deal with study designs, data management, data analysis, interpreting the results, and the communication part in this portion. So if I may introduce you to the biostat subject, we will go with the study design. This is a very first and the basic major domain of this subject. Biostatisticians play a critical role in the design of studies that are conducted to investigate health related questions. Suppose we are working on a cancer patient, we are designing a sort of trial drug, and we start with the basics of the disease, like what is the disease, how it is caused, how it progressed, which type of receptors or structures it is damaging or related to. Then we go for that specific drug. These are sort of open-ended questions that go along with the research. We find out new things, we go accordingly, we modify our pharmacological trials accordingly. So
Hello, Dr.
study
Usama.
design,
The slides are not moving. We still see the very first slide.
it's
Are
not
you able?
moving.
Yeah, we still see the scope of biostatistics and biochemistry, the very first slide. Um,
Still not moving.
Yes, that is what we see. We don't see any other slide.
Can you take it to slideshow, please? Maybe slideshow, then you should be able to move it. Thank you.
there, that's good.
Can you see
That's
it now?
working.
Okay. So I was talking about the study design. Biostatisticians play a critical role in the design of studies that are conducted to investigate health related questions. These are sort of open ended questions. We mainly deal with how this disease started and how it progressed and how it is related to cellular structures. This part of the disease comes in under the biochemistry. But if we start to investigate that health issue, we go with the questions, mainly leading questions, that we start with the very basic question, how it is started, how it is related, how it is going, how it is progressed, and what is the incidence of that disease. This is a main important question of this portion of biostat. The incidence of disease is very important aspect in many diseases, many familial diseases, and sort of other contagious diseases as well. Some people are more susceptible to it, some people are less to it. So these questions help researchers to identify the appropriate study design, select the appropriate sample size, and develop the data collection instruments. So for each study design, we have to choose a proper sample size. Sample size means, for example, working on research based on anxiety disorder in the students. So if I have to choose a sample size, I would choose, most likely choose the students of high school maybe, or college or university, because they are openly able to express that they are having sort of anxiety, or able to express their issue with whatever they are having. So that comes under the sample size, and then develop the data collection instruments. It is basically we develop questionnaires or different type of instruments or tools to assess the condition of, for example, if a patient is having sort of anxiety, as I gave the example of the anxiety issue. So we develop a questionnaire. We ask about their, whereabouts, basics, how they feel when they feel the anxiety, what which thing triggers it the most. So it all comes under the study design. So we have, for example, we design a study. We defined a sample size, we collected it, we were to we for assessment. Now the next part is data management. Next part. So in this part, we the data we already have, we manage it properly. We put it in order. For example, we put in Excel sheets whatever the data we have. We apply the data entry protocols, monitor data quality. We will check if there is any error in the data. So this is the second part. If we go basics from the basics of the biostats from the study design to the next part, the very next step is data management. The very next step is data analysis. We designed a study, we collected the data, we managed it, and we are now going to analyze it. So biostatisticians are variety of statistical methods to analyze data collected in a study. They help researchers select the appropriate statistical test, interpret the results of these tests, and draw meaningful conclusions from the data. So we have a data in a file. This is very in very raw form. We apply different sort of tests. We analyze them. We if some of you from a biostat background, they might know already be with the p value or chi-square test or different type of analysis tests. So this comes under the data analysis. Moving to the next part, interpreting the result. We got the data, we analyzed it, now we have the results. Okay, we have the result file, but this is still in some sort of unreadable language. So biostatisticians help us to interpret their data to put into appropriate, publishable form or to readable form. They potential confounding variables and rest issues related to casualty. Acha, scientists basically form the and to make it possible to very simple language for biostatisticians play a key role in communicating statisticians, policymakers, and general public. They help researchers communicate the results of their studies. So the publishing of that specific research we were working make it easy for the scientists or researchers the major cause or the study they were working to study. It is deployed in many and health field tail of the community medicine, public health, because the programs. So it is useful in determining if a vaccines in epidemiological studies, medical research conclusions about disease in certain population groups. It predicts mortality rates, symptoms, and even biostats in public health to of vaccines and different health. For example, if we are assessing somewhere, then we assess every year if they have any family history or if either it was about their diet, about their lifestyle, everything that includes under the public health portion of which these stats are calculated under the biostats. Okay, so if I give example of the recent pandemic COVID-19, it was definitely everyone knows how disastrous it was. So different people show different type of responses if they get infected or if they got infected. It is because they have different type of immune systems, their genetic makeup is different, some are more susceptible, some are less susceptible. So this kind of factors all come under the public health. Okay, moving to the next part, it is about the cancer research. Cancer is not one size fits all disease. So cancer is more like a dynamic disease uh, due to a very complex etiology, many risk factors, and many things. Because it is widely, it is widely scattered, sort of in different systems. If we dig deep into it, they, for example, if a woman is having a breast cancer, suppose, we don't know. We assess it by ultrasound or mammography. We put her under the tests. We went her through a lot of different type of treatments, but it was all about the gross things we were saying. We are not seeing the molecular differences it is making, or it is going to metastasize at molecular level, because everything is not traceable. We got the receptor combinations of the breast cancer for we got her genomic typing, everything else, but some parts of I still think that some antigens and some other things are still not traceable. That's why it is quite a vague disease. We do not know how or where it is going to metastasize, if it is invasive or malignant. So it is a very aggressive topic under research. So biostats is being actively deployed in this portion. So examine traits of the cancer and occurrences in various ages, genders, and racial groups to work on the prevention. So we cannot assess when a person is going to have cancer, or maybe it's their hard luck or whatever, but we can assess how it is progressing, or where it occurs the most, in which age, in which gender, in which racial groups, so we can work on that prevention. So it is only possible if we have this kind of data. We have all the details or all the homework we do on that clinical trials number based. So we do prevention mainly on hit and trial method. If a cancer is properly preventable, for example, in females the cervical cancer can be preventable if the major causative agent is a virus. If detected early, it can be prevented. So if we know how many women are going to be under screening for that specific virus, how many women are not going under the screening, how many women are going if they are diagnosed with the cancer, if they are not diagnosed with the cancer. So all these stats comes under this domain. In the next portion, we are going to talk about advanced medical technologies. So, as everything is digitalized in this era, so Biostat is more like digitalized. We have latest softwares like SPSS and other a lot of working going on. So this is quite made easy by that computerized systems. So it is also used by the diagnostic procedures or whatever. Computer intensive biostatistical methods, bootstrapping and resampling methods, microarrays, next generation sequencers for genomics, mass spectrometry. There is no there might be students getting confused if they are receiving a lecture or sort of, but this is not something to to memorize it. Just understand what I'm saying, or what is whatever I'm telling, the significance of it, or wherever it is getting used. Just have idea of that. No need to memorize all these things like a proper boring lecture. Just understand it. So it is also used in a pharmacology, as I told, that development of drugs and trials, all the data and the assessment of that data all comes under the biostats. Next, the portion is ecology, constructing biological pyramids, ecological pyramids, calculation of total energy content in the different trophic levels. So if we are studying zoology or more like the having major subject of ecology, we dig deep into it. So biostats is also employed there in different kind of calculations. Effects of climate change and other environmental impacts on ecology communities. This is a very hot topic for the research these days, especially due to a lot of floods going on, earthquakes going on, and other natural disasters. So the next part is demography. So it is biostat is used in the estimating the attributes of the population, sex ratio, birth rate, density, used in drawing growth curves of the population at the given time. Logistic growth curve. It is a type of curve to show population. So if we do census, we do analysis of different type of diseases occurring in a specific region. It all comes under demography. Birth, we are, for example, calculating the birth rate, death rate, maternal mortality rate, or any other specific epidemic, or if we are uh, mainly calculating it in a close or a specific geographic area, we count it in under demography. So biostat is deployed in that calculations as well. Population genetics and statistical genetics, it is almost same thing. Link variation genotype with the same variation, with the variation in the phenotype, study distribution and challenge in allele frequency. So we basically deal with all sort of genetic changes, variations, and the variations that tend to cause familial disorders and the cancers and their incidence. Everything we see it under this portion. Bioinformatics, in biological sequence analysis, assign function to genes and proteins by study of similarities between compared sequences. In bioinformatics, basically we closely zoom in the structure of a DNA or RNA, their sequences and their central dogma or anything. We do analysis, especially sequence analysis. It is under bioinformatics. So bioinformatics is also closely linked to biostats. Systems biology. Systems biology is a biology based interdisciplinary field of study that focuses on complex interactions within biological systems. It makes heavy use of mathematical and computational models. So in the next, probably the heading should be agriculture. It is by mistake. In the improvement in agriculture works, the decision as to whether one variety of crop is better than the other will be made the results of a careful planned series of the statistical experiments. So in agriculture we also deploy statistics because we with the need of grain or increasing population, so we want to grow better crops with better yield and more money. So with a better seeds, we estimate the production of a specific crop, that if previous seed was better or the new one is better or the modified one is better or the classical one was better. So it also, the statistical part comes in under biostats. So moving to the next part of biochemistry. Biochemistry, as its name says, bio plus chemistry, biology plus chemistry. It mainly deals with the biological structures at cell micro or macro level. Biochemistry is a study of chemical processes and substances that occur within the living organisms. Okay, we starting from the very basic structure of a cell organelle, or even cells genetic components, to its metabolism, that this all comes under the biochemistry. It focuses on the chemical basis of life, including the structure and function of biomolecules like proteins, nucleic acids, carbohydrates, and lipids. Biochemistry plays an important role in biological and medical research as it provides insight to the molecular mechanisms that govern biological processes. So, if we know the normal processes going on, we can only trace the pathological process. If we don't know the normal functioning or structure of a cell, we do not know whatever it is going on in the cell, whatever disease, specific disease is. So if we know the normal structure of the cell or its molecules, then we can only be able to trace its abnormal if the disease progresses or a disease starts. So we can properly tackle the disease according to its structure, its mode of uh, occurrence. Speaking of the scope of biochemistry is broad and encompass various fields including medicine, agriculture, biotech. So it is in the medicine, it is mostly used in the molecular level. For example, to develop new drugs. So to develop a new drug, we have to make a proper receptor or drug combination. So if we know the receptor structure of the cell properly, we can only design a drug according to that one. Okay, if we do not know the structure, or we if we're blindly applying drugs, they go haphazardly anywhere, or if they are not responding properly, we need to know the cell structure, cell membranes components, or whatever proteins in it, whatever receptors in it, to develop a drug. Similarly, if we in medicine, speaking of the blood tests, serology, CBC, or speaking of LFTs or RFTs, there is a major role of biochemistry as well. For example, biochemistry is used to study the structure and function of proteins and enzymes involved in the disease pathways and develop drugs that can target these molecules. Biochemistry is used to develop diagnostic tools that detect disease biomarkers such as DNA, RNA, and proteins. So, biochemistry is also deployed. For example, I will give another example of breast cancer as well. In breast cancer, there are certain type of tumor markers, as I said. Here is the disease biomarkers that specify the specific type of cancer. We can only able to give proper treatment that fits properly to that type of cancer. It is only possible if we knew biomarkers of that disease. For example, estrogen receptor, progesterone receptor are most common on the breast cancer tissues. So if we run a biopsy on that tissue, we get to know that these biomarkers are present. Okay, we have these kind of drugs available. We can apply or give to patient, administer to patient. We can put her on that chemo or that radio. This is going to affect that way. So the pathways, mechanism of action of drugs, and how it is going to affect at cellular level, this all comes under the biochemistry. Next is the applications. First one is the drug development. Biochemistry plays a critical role in the development of new drugs. Biochemists study the structure and function of biological molecules such as proteins and enzymes to understand how they interact with drugs. Okay, there are different types of tons and tons of drugs that act differently. Some act as a membrane, some goes inside the cell, act at the on the nucleus. Some act as on the enzymes, specific enzymes. Some bind just go bind and bind to proteins and show their effect. So each drug has different mechanism of action, and if we knew their specific biochemistry, how they are going to act, or which structure is they are going to bind, which enzyme they are going to inhibit. So this is very important, and this should be this much precise to cure a specific disease. Next is a clinical diagnosis, how we can use biochemistry in clinical diagnosis. Biochemistry used in clinical diagnosis to measure the levels of biomolecules in the body, such as blood glucose, cholesterol, and enzyme. This is a very basic example that we, measure blood glucose level, HbA1c, cholesterol, enzymes, different other blood tests. We, for example, check liver functioning tests. We check renal functioning tests. This all comes under the domains of biochemistry. We are dealing with the biological molecules. We we detect those variations in the serology or hematology if we check the sample. So for example, if a person is having malaria or other disease, in which random, we suppose a random disease in which his, his platelets are going are rapidly declining. He's having nosebleeds, he's having rashes on the body, he's you know vomiting blood and everything going on. We don't know what's wrong with him. So we decide to check his blood. We decide to do a CBC, complete blood count, on that person. We get to know that he's having less platelets than the normal percentage. He's, for example, if he's anemic, he's having less red blood cells. So this all comes under the clinical biochemistry. These measurements help in the diagnosis and management of various diseases and conditions. Some tumors, shed their biomarkers in the blood. We can detect them by blood. For example, if a person is having lymphoma, we will if we do CBC, we will check there are a lot of wandering lymphoblasts around. If a person is having sickle cell anemia, we do HB electrophoresis. HB electrophoresis is also, you know um, technique in the biochemistry. So if we do HB electrophoresis, we can detect the sickle cell anemia. So this is a very important diagnostic tool that biochemistry is being employed and actively used in the diagnostic procedures. Next, agriculture. So biochemistry used in agriculture to develop new crop varieties that are resistant to disease and pest. This is a major issue. Sometimes there is outbreak of diseases or pests that eat up all the crops, and this causes a widespread economical loss as well as. So to avoid that condition, to avoid the pest to be disease resistant, some scientists starting to develop or started to develop specific genetically modified seeds that are resistant to pests or resistant to diseases. So this was only possible to because of the biochemistry. If we knew the normal structure of those, genetic makeup of those plants or those crops, we were able to identify which gene or which allele is making them susceptible to that disease or that specific pest. Or so we were able to modify that specific gene, or we were do some minor changes in the genetic makeup to make them giving more yield and to be more disease resistant and best. So it comes under the agriculture portion of that we are going to utilize biochemistry here. So biochemists study the biochemical pathways in plants to understand how they respond to different environmental conditions and develop new strategies to improve crop yield and quality. Okay, next comes the food science. Biochemistry is used in food science to develop new food products, how they are metabolized in the body, to understand how they affect health. Okay, in the food science, there is a lot of adulteration going on in the local food streets, or in third world countries. There is a lot of disease related to the food poisoning. There is a lot of gut disorders related to the food, because people mostly get infected. Either they get typhoid or they get H. pylori infections. So it is very important to check and balance the food circulating in the streets or in the markets, to properly check if they are good to go, or if they have proper expiries, or if their shelf life is longer enough, or if they are safe to use. They are no they are not containing any carcinogenic or other sort of additives that are potentially harmful. So this all comes under the food science. We check the quality of the food, we check the chemical composition of the food, and we check how their metabolites are going to excreted from the body. If for example, if a food is seems okay, it is not harmful, a person eats it, and the food's metabolites or its nanoparticles or whatever it is, after its metabolization, uh, it causes some sort of disorder or disease, or develops a guts disease in the person. So so that specific food is not fit for the market. So we have to make sure that food is good, that food quality is good, food doesn't contain any specific additives that are carcinogenic or make cause potential harm, in immunocompromised persons, in the pregnant females or in the children, or if they have their quality is good. So this is a very detailed or a very vast subject, food science basically. So we deal with the biological molecules mainly here as well, the chemical composition of food and how its metabolites are going to react. Next is the biotech. In biotech, biochemistry used to study and manipulate genetic information. So if before I go in its detail, I would like to give an example of antibiotics. In the biotech, how we produce antibiotics. If any of the students know, can you please respond? Can you hear me?
Yes, we can hear you and we can see the presentation just fine.
Okay. So the antibiotics are basically developed from the bacteria or their plasmids. We know the structure of bacteria, that specific bacteria. We manipulate their genetic information. We put into some we you know uh, kill its virulence. We use its antigens to develop vaccines, to develop immunization. So we use biotech for the for making a more virulent strain into less virulent strain. Or for example, uh, during these days, for example, there is a major issue of bacterial resistance against antibiotic. It is very hot topic under research as well. So how we are going to cope up with the bacterial antibiotic resistant, because there are this is a very fast growing condition, and it is very very it can be potentially harmful if there is no antibiotic left that can cure the antibiotic resistant strains. We have to manipulate, we have to find a way that how we can stop that specific bacteria who is not responding to any of the available antibiotics. So if I give a raw example, there are specific viruses that are called bacteriophages. We can manipulate their genome, or we can make them infect bacteria that are antibiotic resistant. This way we can only kill that bacteria, specific bacteria, because no antibiotic is ever gonna kill them. So we are manipulating the specific viruses that infect bacteria and kill them. So this is a very classic example of how we can manipulate the microbes to for the different diseases. In biotech, we develop different we have different developments, specific productive developments in crops, in food, in animal husbandry. We develop different type of animals with better genetic makeup. For example, we develop different cows, different type of cows that yield more milk, that have that are disease resistant or have better survival. So this is all about genetic manipulation, how we are able to check and replace or amend the susceptible genes. So biochemistry used to study DNA and RNA and develop tools for the genetic engineering, such as gene editing and gene therapy. So gene editing is basically more like we find a gene, it is we find a gene, it is faulty, we identified it, replace it with a normal, or maybe if it is excessive, or there are multiple copies of that gene, there are multiple ways to develop to cure this disease. For example, gene therapy is actively employed to cure different type of blood cancers. So for example, biochemistry used to develop new biopharmaceuticals such as recombinant proteins and monoclonal antibodies, which are used to treat diseases like cancer and autoimmune disorders. Autoimmune disorders is also a very dangerous condition in which a person develops sensitization towards its own body, its own receptors start to attract antibodies of the its own. So it's more like uh, damaging its own. There are a lot of diseases like this. To cure autoimmune disorders and the cancer, monoclonal antibodies are also being considered a lot these days, specifically for the treatment of hepatocellular cancers, advanced cancers. Monoclonal antibodies are being used on trial basis because they apparently seem effective and no other choice. So we are going for the developments, and every day we make new changes or make new monoclonal antibodies or proteins that are getting every day. We if we go 10 years or 20 years back, there was no though advanced treatment like this we have today. Maybe after 20 years in the future, we might be able to have even better treatment for the cancer, or we might be completely cured, might have complete cure. We are making in the biochemistry and the biotech. This this fields these two fields are also closely related. So biochemistry is also used to develop new biocatalysts such as enzymes and biofuel production. So this is also a big application of this one. So as we are running off fossil fuels as well, so in biofuel production, microbes are also being employed by remediation. We also using microbes for the to overcome the overcome the pollution, and the water pollution we are having. So biocatalysts are enzymes, and microbes are basically we are using these in the bioreactors of to make various type of drugs and other developments in the medicine or the pharmaceutical industry. There are a lot of career prospects regarding both of these, but main one is the researcher. Research is the basic very thing. Research, if we talk about research, because every new development or every new thing starts from a researcher. If we develop a new drug, or if we do a new discovery of the disease, or make any significant improvements of the pre-occurring diseases, so the very basic, the center point is researcher. So there is a very good opportunity after these two programs, that a person may go after his post graduation or PhD into research of different renowned institutions. They become food analyst. This is also very important field, agrochemist. So these three are mainly related with the various aspects, with the cure of the diseases, with better production of the crops of the animals, of to be disease resistant, to give more yield, to give more good quality of food, for better treatments of the diseases, for a better quality of life. So these three are the main uh, career prospects for these two subjects. Plus there are a lot of them more, but I didn't mention it here to cause any further confusion or students getting double minded or sort of anything. If I summarize this, in the modern day medicine, this relationship between biochemistry, biostats, and medicine is integral to advancement of medical knowledge and the improvement of patient care. So, coming from a medical background, I would highly emphasize on this part, that the biochemistry and the biostats and the medicine, they are kind of inseparable for the better development of a treatment, or a strategy for, or a treatment guidelines, or better patient care. So discoveries in the biochemistry are leading to the development of new therapies that are more effective and have fewer side effects. So in this point, we are going to talk about that there are a lot of drugs that we already developed, but they have, you know, very serious side effects or have adverse effects. Adverse effects are very rare. They occur in some patients, but they are lethal. Side effects are more common, like nausea, vomiting. So we are planning to develop drugs that have least side effects, that are more disoriented, that's they are going to cure. So this discoveries, the ongoing discoveries and the research is also related to these fields. Biostats is essential to in the design and analysis of clinical trials that test safety and efficacy of these therapies. Okay, we have developed a new drug, we got under it trials on the lab animals. We uh, we did it, tried on the clinical trials, we got it approved, but there are still a lot of reservations regarding it, unless some patients report adverse side effects that are, that scientists doesn't calculate in their theoretical when they are developing, or never have checked it before, or encountered before this kind of effects. So this is an ongoing process of developing new drugs, new and better drugs than before, that they already developed, more specific drugs that target the specific disease instead of targeting other normal parts of the body. So for example, in the cancer, there are a lot of advancements. For example, we have localized chemotherapy, the radiotherapy is also localized, but we are manipulating the radiation radiotherapy as well to cause less damage to the skin and the normal tissues around. We are making it more well targeted to the fast growing tumors, and we are sort of implanting different type of chemotherapy releasing tissue implants that are inside of the skin, or different, there are a lot of different developments going on. So this field isn't never going to to get dull or dead sort of. This is a very active and uh, under research area. So, in summary, the relationship between biochemistry, biostat, and modern is critical to diagnosis, treatment, and prevention of the diseases. Advancements in biochemistry and biostats are leading to the development of the new therapies and the interventions that improve patient outcomes. And the integration of these disciplines with the medicine is essential for advancing medical knowledge and improving patient care. So, every day we make new progress. The advancements in the biochemistry and biostats are telling of us the results going telling us about the trials going on, about the new discoveries of the receptors or the new antigens and the new drugs that are going to target. So their efficacy is being calculated in the biostats. If they get approved, it comes into market, then patients report the side effects or adverse effect that previously wasn't noted, or it wasn't checked or anything, that then the drug again undergoes a procedure of rigorous testing that to minimize those side effects. So this is an ongoing process. Thank you, that's all. If anyone of you have questions, please ask. Okay, I have questions in the inbox. Let me read one by one. Tom Thompson is asking me to share my slides. Okay, I will share you my slides file. Next, so much research you will need to methodology. Good topic for presentation.
Well,
Thank you
I
so
think,
much.
thank you so much, Dr. Usama Jamil. So let's go to some questions here. TechnoSpark. I'm going to ask you to unmute your microphone. TechnoSpark 2.
Okay, sure. So I can't hear you.
If we can't hear you, you should write your question into chat.
If any questions, please you can chat. I am getting the connection right now.
Yeah, TechnoSpark, write your question into chat. We're going to go to Tangavel Kopen.
Hello, thank you, sir. Thank you for giving the opportunity to ask a question to Dr. Jamil, who has done a nice presentation. Yes, doctor, do antibiotics damage our body, and can it damage us? Can you listen to me?
So the question is do antibiotics harm
Damage,
the body?
yeah. And can it
No,
damage us?
actually, most of the antibiotics have very
I can't hear anything.
I would suggest let's stop the presentation.
antibiotics very well, and we have to.
Okay, Dr.
So
Usama, try to speak now.
antibiotics properly prop and for the proper and the proper disease. If anyone is okay, can you hear me?
Your audio is like cutting out and
So
coming back on.
antibiotics to anybody, that proper are going to use it, unauthorized or unprescribed.
It's a really interesting question, but
Is it clear
the
now?
answer is not coming through clearly because of the audio.
Is it clear now?
It is clear, yes.
Can you hear me, sir?
Yes,
Now,
much
Yeah,
better now.
much better. Yeah, I agree.
So Dr. Osama, please start the
that
answer
and
from the
doesn't
beginning.
harm unless they are excessively used, or if they are a disease for a specific condition. If the antibiotic is being used in a for a disease for a proper day, it is not going to harm the body.
We can't hear anything here.
Usually the antibiotic is given in proper for like 3 days or 5 days. You need to cut it down. It is not going to cause any side effect. Your connection is
Yeah, it's hard to hear the full answer. It keeps cutting out, but we're getting an idea that if you have bacteria in your body, it will help. But if you, but also the antibodies will do their job too. Is that correct?
audible.
Yeah, because many people say that the modern world is using
Yes,
antibiotics
where
too
we
much.
antibiotics. If it is not by itself, sometimes we yes, it is
Doc,
right, but we don't antibiotic time. Our immune is healthy immune system. He doesn't always have to take antibiotics, and not every infection resolves on its own in a day or 2 or 3. infection persists or goes for severe infection, so we can administer antibiotics, but for a very specific, for a, very precise dose, then it is not going to cause any effect. Is that clear now?
So basically is sometimes the body does not have the antibodies, so then antibiotics help the body because there's a deficiency in antibodies. But and he's saying that antibiotics should not harm the body. Okay, let's go to
Yes.
Moji
Oh,
Sola Esther.
antibiotic.
We have about 2 or 3 more minutes, so we only have room
Yes.
for maybe 1 or 2 more questions.
Sometimes we don't have
Dr. Usama, you may consider writing your answer into chat.
Okay, please ask.
Okay, Mojisola Esther.
Okay, thank you very much. Thank you so much, Dr. Osama. Thank you. Okay, 2 questions. The second one, you partially answered it.
Next question,
But the
please.
first question is, I'm looking at genetic mutation in animals, plants, which is somewhat what biochemistry is also speaking to, because it's also used to come up with new plants, new drugs,
Yes.
and new scope of things. My question is, all these mutations as against organic growth that we know, don't they affect the health of human beings? That's number 1 question. The second one is, how sure are we on the safety of all these things, all these different animals and variety of plants and all of that we are getting from all those different simulations, mutations from biochemistry? What's the safety level? How safe are we?
Okay.
Thank you.
The mutations in the other animals. Can you hear me now?
Yes,
can you
we
hear
can
me
hear
now,
you.
miss? I will answer you now. Okay. The mutations in the other animals in the plants doesn't affect us directly. However, if the crop isn't good, or it is not, yielding anything, we are going under economical loss. So we will, tend to use genetically engineered seeds to, overcome that thing. Mutations in the animals, sometimes we, uh, we use biotech or biochemistry. We do genetic engineering on animals as well, to to make them yield more milk, or make them more beautiful, or sort of, in case of pets, if we're talking about, or in case of, if we talk about livestock. So for better meat, or for better taste, for better quality of milk, we do genetic engineering. So mutation doesn't affect us directly, but they are affecting us indirectly by economical ways or other ways, because we are too much relying on them as well, on the crops. We are, for example, economically dependent on crops. We depend on the crops feeding for animals. If we get better animals, better meat and better milk for genetic engineering, then why someone prefer the classic way of animals or
Okay, that is all we have time for in this presentation. So I guess on that note, is if you're doing genetic mutations, there's a danger if you create poisons for the body through the genetic mutations. But if you create better resistance to the weather changes going on in climate change, that's a good thing. But if somehow you create a poison, that's a bad thing. So you have to do research to find out if what you've created is poisonous or helpful. And I guess you go much deeper into that. Another issue with genetic mutation is you don't want to create a culture of just a few genetic variations, like in the agriculture of the United States. There's not much genetic diversity, and that's bad, because as things change, you could have severe changes in crop production. So just a few things there. But I want to thank Dr. Usama Jamil Mohammed for a wonderful presentation on biochemistry, biostatistics. This is really important information that a lot of you can use in your own research. And we strongly suggest that you learn statistics, you learn how to do a research design, and then do data collection, and then find the appropriate test for analyzing your data. It's not that difficult. The statistics don't have to be 10 pages. You can do a statistical test in 2 or 3 pages, or even 1 page of your assignment, or even a thesis. It doesn't have to be complicated. It just has to follow if you have the correct statistical test, you've done 100% of your work, really, basically. That's most of what you need to do. And so keep it simple, keep the statistics simple and correct. And it doesn't have to be complicated. And you'll get good results. You'll get good data that you can analyze. So I'm trying to look. I think Dr. Usama Jamil, you're still here. I don't see you. Where are you? But, oh, there you are. And so I want to thank you for your presentation. Is there anything more you want to say? I'll give you a minute left for anything you want to say.
Okay, I was answering to that. If you can hear me, I can, answer the very first question. Coupon asked. Okay, it was about the antibiotics, right?
Yes, about the antibiotics.
So if a person is having mild infection, he doesn't have to take the antibiotics. The infection usually resolves on its own. If the infection persists or the infection is taking too long to recover, he should take antibiotics, but with the prescription for the specific time doctor prescribes him to. Okay, and the antibiotics doesn't necessarily harm anyone. It may usually cause very mild side effects, may nausea, vomiting, sort of. If you have any adverse side effects due to any antibody, you should discontinue right away. Okay, so they doesn't harm anyone. They are basically support our immune system to fight against that bacteria or that virus. You got my point?
Perfect.
Okay.
That's all the time we have, so we'll have to end the live presentation now.
Is there anything else or any question?
Nope. Thank you, Dr. Usama Jamil Mohammed, for a wonderful presentation.
Thank you.
If you have another subject to talk about, please come back and we can arrange another presentation with you. This is very important information for everybody to learn. So
Yeah, sure. I would love to.
Okay. So everybody, that will do it for this live session from Dr. Usama Jamil Mohammed about biology, biostatistics, biochemistry, and how to develop your experiments and what areas like food science, agriculture, cancer research, genetic mutations, all these types of areas, pharmaceuticals, that you can use this research biostatistics for. So thank you so much.